I'm Dave Plachetzki, and my lab from the University of New Hampshire is interested in understanding how senses evolve and how they originate. So we take sort of a much broader macroevolutionary view than a lot of the talks that we've seen so far today. So hopefully this talk will serve as sort of a complement to that great microevolutionary work. I'd also like to point out that I think I was a little overambitious with my talk title. So rather than talking about our current ideas about a ancestral sensory toolkit in metazoans, I'll talk really just about some things that we found with chemosensitivity recently. So humans or animals interact with their environments through the use of a set of diverse extra-appropriative senses. So we're humans. These are the Aristotelian senses, if you will. These are the senses that Aristotle defined. But of course, the human senses have a much deeper and more diverse evolutionary history. So if we were flies, we might think of these as the senses. Flies also have similar, either homologous or analogous modes of sensation. And one of the big questions that we're interested in trying to understand in my lab is what are the evolutionary relationships between the senses amongst, broadly divergent taxa that maybe split 600 million years ago, such as humans and flies. Our taste repertoires between vertebrates and insects or earlier animals, are they homologous or are they analogous? And so these are sort of the questions that we're really interested in understanding. And so today, I'll talk about how we use whole genome phylogenetics as a tool to address these questions. And we've been working on all of the known sensory receptors, and there's a growing handful of these. But today I'll talk really just about two different types of chemoreceptors. And we can think of these two different types of chemoreceptors almost as an axis, if you will, across animals, across metazoans. So there are taste 1 receptors. These are what humans and other vertebrates use to interpret sweet tastes like sugars and amino acids, savory tastes. And I'll talk about some new work that places the origin of taste 1 chemoreception actually very early in the evolutionary history of animals. And one thing that comes out of that work is that these taste 1 receptors are actually lost in a major group of animals called protostomes. Protostomes, which I'll explain in a second, include things like insects and nematodes and other invertebrates. And then finally, I'll talk about the origin of a group of taste receptors called gustatory receptors, which are known from protostomes. And we will argue that these are actually present much earlier in the evolutionary history of protostomes than presently thought. And I'll introduce this hypothesis, what I call a chemosensory paradigm shift, from sort of a GPCR, a G protein coupled receptor, taste 1 repertoire to a gustatory receptor based system in protostomes. Okay, so one of the tools that we use to understand how gene families evolve is actually the animal phylogeny. So last year, this came out I think last October or something, maybe January, I don't remember. This paper was published last year, and what we did was we took as many whole genome data sets from animals that represent the major diversity of animals. And we put together an alignment that had about 1,100 genes in it, and we filtered that alignment extensively in many different ways, and I don't want to talk about it too much, it's published. But essentially what we came up with is this fairly well-supported tree. I'll call it well-supported. We can argue about it, but hopefully we won't argue about it today. And I would point you to many other papers who have found this result. The key here is that this is a tree of animal genomes. So we can make gene trees from each of these genomes represented here. So not only is it interesting and highly illuminating to think about the animal tree of life, but it's also very interesting to have an animal tree of life represented by whole genome sequences. And so we use this phylogeny really as almost a pipeline for understanding gene evolution. And so we can search through all of these genomes, and I won't speak too methodologically here, but we can put together fairly exhaustive sets of gene families for any gene that we like. And we can use other sorts of approaches to correct these trees, these gene trees, based on what we think we have pretty good evidence for as being the species tree. So we can correct these gene trees with algorithms like TreeFix, which work pretty well. And then finally, we can map the origins of any gene family onto this phylogeny. And so today, this is really what I'll be doing with chemosensory gene families. Okay, so first off, these are the taste 1 receptors. These are what humans and other vertebrates use to interact or to taste sugars and amino acids. One thing I'll point out about taste 1 receptors is that these are G protein coupled receptors, and that means that they interact with other proteins like G proteins to elicit their signal. So a taste 1 receptor alone cannot induce signaling, much like opsins alone can't induce signaling. There's required some other transduction mechanism. Another thing I'd like to point out is that all GPCRs have their N-termini exposed to the extracellular realm, whereas the C-terminus is buried intracellularly. Okay? And again, we use taste 1 receptors in vertebrates to detect amino acids and sugars. Okay, so when we apply our phylogenomic approach to taste 1 receptors, we wind up with something like this. So first of all, I'd point out that the taste 1 receptors that we are familiar with are actually part of a much larger clade of chemosensory receptors. So the biggest clade of all of these are actually vomoral nasal 2 receptors. So taste 1 receptor, oh, and this is all known so far, this is nothing new. Taste 1 receptors are part of a much larger gene family that includes vomoral nasal 2 receptors. But they also include calcium receptors and these things called GPCR6 receptors. And to make a long story short, both calcium receptors and GPCR2 receptors are also involved in chemical sensing. They're also actually involved in tasting in the vertebrates that have been studied. I'll point out that the branches here are colored based on the taxonomic groups, roughly the coarse taxonomic groups that these taxa are from. And so if we go deeper into the tree of this clade that includes taste 1 receptors, we have a large clade from Cnidarian taxa, shown in red, and then some other sister, or actually not sister, but another clade from Plachetzzoans, trichoplax, and then some other even earlier branching representatives. And so we interpret this as really meaning that this clade of genes that includes taste 1 receptors is actually a really ancient chemosensory receptor clade, and that Cnidarians and Plachetzzoans and other earlier branching metazoans also have these genes. They have orthologs of these genes. Now, one thing you'll point out, if you just look at the pattern of these different clades, you'll note that there are no green clades, there are no green branches, that is, in any of the taste 1 receptor gene family. But the sister to taste 1 receptors are metabotropic glutamate receptors. And here we do see that for every blue clade, it's sistered by a green clade. And this really represents the relationships between protostomes and deuterostomes in metazoan phylogeny. And the other thing that it means is that there are no taste 1 receptors present in protostomes. There aren't any. They just don't come up. And we've looked really hard, and we think we found snippets of them in some genomes, but I'm not going to talk about that too much today. So if we take this result from taste 1 receptors and we map it back on to the phylogeny of metazoans, really what we can say is that taste 1 receptors, or at least the gene family that includes taste 1 receptors, and the gene family that we infer ancestrally was chemosensitive, dates back to the origin of animals themselves. So taste 1 receptors, even though we think of them as sort of a vertebrate innovation, are actually part of a much deeper evolutionary lineage of chemoreceptors. And if we have time, I'll show you what we think they're doing in these earlier branching taxa. Okay, so that describes taste-1 receptors. But I noted that protostomes lacked these altogether, and that we would infer that they were lost. Well, that doesn't mean that protostomes can't taste sugary things. Of course, we were just watching some butterfly protostomes tasting sugar water. So what insects use for tasting sweet things are called gustatory receptors. And one thing that's interesting about gustatory receptors, even though they've got this transmembrane, 7-transmembrane domain architecture, their C-termini are extracellular, and their N-termini are intracellular. And these are not G protein receptors at all. In fact, they function as ion channels. So gustatory receptors act directly to modulate a cell physiological response to some environmental cue, whereas taste-1 receptors need G proteins and all that other stuff. So we can sort of compare the architecture between gustatory receptors and taste-1 receptors. So the question becomes, when did gustatory receptors evolve? We know that they're present in insects. How deep do they go? Well, if we do the gene tree of gustatory receptors, we see that, and I'm just labeling these by color, but a class of animals called lophotrophozoans, which includes annelids and mollusks and other protostomes, in addition to nematodes, chelicerates, myriapods, crustaceans, and insects. All of these guys have gustatory receptors, or at least orthologs of gustatory receptors. Maybe they deserve a different name, we'll see. And so what this means is that gustatory receptors go all the way back, at least to the origin of protostomes. And I guess we could argue about whether they go deeper. So in summary, what it looks like to us is that ancestrally, the first animals, or at least the last common ancestors of extant animals had taste-1 receptors, and that those were then lost in certain lineages. But at least in terms of protostomes, those were potentially replaced organismally and ecologically by gustatory receptors. So this is what I mean when I say a chemosensory paradigm shift, because we're really going from a G protein coupled receptor based modality to one that's driven by ion channels. And if I can hurry, in the last 30 seconds, I'll tell you that in cnidarians, it looks like cnidocytes, these stinging cells, are actually being controlled by the orthologs of taste-1 receptors, and we can modulate their stinging behavior based on the presence of amino acid cues and sugars in the water column. And so with that, I'll just summarize. It looks like taste-1 receptors were present very early in the origin of animals, and that they were then lost in protostome genomes. And we are thinking about this as being indicative of a paradigm shift relative to how animals interact with the chemical environments. And finally, I just mentioned very briefly that taste-1 receptor orthologs from cnidarians seem to be functioning in modulating cnidocyte or stinging cell behavior, and that's what cnidarians used to eat. So with that, I'd like to thank everyone from my lab at University of New Hampshire, especially Molly Hartley and Heather, who did a lot of the in situ work that I didn't talk about very much. And I'd just like to point out that this work really has its origins in my postdoc and even back to my graduate days. So thanks.